revision session on photosynthesis and how that links to the structure of chloroplasts. So let's have a little think about, first of all, the structure of a plant cell. So a plant cell has got around it a plasma membrane, okay, and we would call that the cell surface membrane. That is surrounded by a cellulose cell wall. Now normally we can't see the cell surface membrane, so often we could draw it like that to show that the membrane is pushed right up against the cell wall. Inside the cell surface membrane is cytoplasm, and I'm just going to draw that with a few little dots to show other organelles that might be present, such as ribosomes, mitochondria, ER, things like that. But of course, we wouldn't be able to see those organelles under a light microscope because the resolution is far too low. One of the biggest organelles that you can see with a light microscope would be the vacuole. And we're not going to talk about the vacuole at all today. Another big organelle that we would see, of course, is the nucleus. And we're not going to talk about the nucleus very much today either. So this here is the cytoplasm. And because we're talking about photosynthesis, an important organelle that we would see inside the cytoplasm of a plant cell that was exposed to light only. So you wouldn't see these in the root cells, for example, of a plant, or the cells that are right in the inside of a trunk or a stem, for example, are of course the chloroplasts. And that is where all the action is going to take place that we're concerned about. And of course, chloroplasts contain chlorophyll. Okay? So what we're going to do is to have a look at one of these chloroplasts. And I think the structure of chloroplasts is pretty straightforward. It's not too difficult. Okay? But what people do have a difficulty in is working out where the chloroplast starts and where it ends. Okay, so we're going to look at this chloroplast here. And the first thing we can say is that chloroplast is surrounded by two membranes. So there's one membrane, and then surrounding that is another membrane. Now, what is important is that you realise that this chloroplast is sat in cytoplasm. So this cytoplasm that we've got here is this surrounding the chloroplast. The chloroplast is made of two membranes, and these membranes form an envelope. Outer and inner membrane. Inside the chloroplasts is a liquid, and that liquid in some ways is similar to cytoplasm but isn't. It is different, so we call that liquid the stroma. Do not get that confused with cytoplasm, which is outside. The biggest structures that you can see inside a chloroplast are the thylakoids. And each thylakoid is a flattened sac made of a plasma membrane that goes around the outside. So in 3 dimensions, this would be, from the top, circular, but it would have a flattened shape. And so that there we call a thylakoid. And what happens is that we have a number of these thylakoids stacked on top of each other like that. And when we have a stack of thylakoids, we call that a granum. And in a chloroplast, there are usually a number of grana spread around. So I'll just draw 2 or 3. But what happens is some of these thylakoids start to grow out and they form long sheet-like structures which can stretch to other grana. And these can be multiple, like this. And they're called intergranal lamellae. So this is one lamella, and the plural would be lamellae. Lamella basically means a sheet. And then there are some other things that are a bit exciting and not exciting in the stroma. Excitingly, we have a bit of DNA. Not as excitingly, we have some lipid droplets. We have some ribosomes. And these are smaller than ribosomes that are found in the rest of the plant cell. And then we'll also have some large, usually some large starch granules. Okay. And I'll stick another couple of thylakoids on the top of that granum to make it look more what we would expect. So how does this relate to the biochemistry of photosynthesis? Well, it's all to do with the thylakoids, because in this membrane that is going around the outside of the thylakoid, we will find chlorophyll. Okay, and this is where the light dependent stage or reactions take place in the thylakoid membrane. The light independent stage takes place in the stroma. Of course, we sometimes call that the light independent reactions. We also have in the thylakoid membranes the process of chemiosmosis. And so the next part of this video is going to show what the relationship is between the structure of the chloroplast, the processes of the light-dependent reactions, the light-independent reactions, and chemiosmosis. Now that we've looked at the structure of chloroplasts, what we want to do is to see the different parts and processes of photosynthesis and how they relate to what actually happens in the chloroplast, to the structure of the chloroplast. Now, before I do, what I'm going to do here is to get a Post-it note, and I'm going to remind myself on here of two important processes. And these two processes are oxidation and reduction. So these are chemical processes, and we use these to describe what is actually happening to the various molecules and substances that are present in photosynthesis. Okay, so remember that oxidation is the loss of electrons. It can also be described as the loss of hydrogen or the gain of oxygen. And we don't often use that in photosynthesis, that last one. Reduction is the opposite. So reduction can be the gain of electrons, the gain of hydrogen, or the loss of oxygen. Now, in photosynthesis, we really talk about electrons and where they are moving. So we're going to concentrate on what we think about electrons. So having a little Post-it note like that, just to remind yourself of what is happening, is pretty useful. So I'm going to take that away and I'm going to place it at the side. And whenever I need to think about what is happening in photosynthesis, I'm going to bring that back in just to remind myself of what is happening. Okay? So if you remember, there are two parts to photosynthesis. There's the light dependent and the light independent reactions. And then together with that, there's this process called chemiosmosis. So what I'm going to try and do is a summary diagram showing where things are happening, what is happening, rather than focusing on just one little process at a time. And we're going to start off by looking at what we just had a look at, and that was the structure of, here we are, chloroplast, and here we've got the thylakoid. So there is one thylakoid, there's another thylakoid making up a granum. Let's just concentrate on this one thylakoid, and in fact, let's just concentrate on that one particular area, the top. So we're going to zoom in and look at the thylakoid membrane, and we're going to look at the stroma and the lumen of the thylakoid, the space inside the thylakoid. Okay, so remember the thylakoid is a large flattened sac. So let's first of all draw our thylakoid membrane. And the thylakoid membrane, and if we do about here, it could be like this. And I'm going to give it some thickness so that we can put some substances in there. So I'm only going to draw a few phospholipids. But like all plasma membranes, it is made of phospholipids. Okay, so this here is the thylakoid membrane. I'm not going to draw the other phospholipids along there because they'll just get in the way of what we're trying to do. What is important here, and we'll state this now, is that the thylakoid membrane is impermeable, so it won't allow hydrogen ions to pass through. And that's really, really important. So here is the stroma, and here, this space here, is the thylakoid lumen. And the word lumen means in the space inside a structure. Okay, so we know in photosynthesis that there are chlorophyll molecules, and those chlorophyll molecules are found in this membrane. So there is a chlorophyll molecule, and we know that that absorbs light energy. So if we have light coming in, it's actually a photon of light, but there is light energy, and that light energy is absorbed by that chlorophyll molecule, and that energy is passed to one of the electrons, or a pair of electrons, in that chlorophyll molecule. When those electrons get that energy, they become excited or energized, so much so that they leave the chlorophyll molecule. Now they can't just go anywhere, because electrons floating around in the chloroplast could cause lots of problems. So instead, there's a protein in this membrane. Remember, the fluid mosaic model is proteins embedded in a sea of phospholipids. So one of the proteins here acts as an electron acceptor. Okay, and the electron moves from the chlorophyll molecule to that acceptor molecule. In doing so, it makes the chlorophyll molecule positively charged. And we call that process photoionization of chlorophyll. So all that means is the chlorophyll molecule absorbs light energy that is passed to the electrons, or some of the electrons in the chlorophyll molecule. They get excited, they leave the chlorophyll molecule, and because electrons are negatively charged, the chlorophyll molecule becomes positively charged. So it's photo, using light, ionization, producing the charge of chlorophyll. Now, we know in photosynthesis that water is split, and that is because of this reaction. Water molecules on their own will split into electrons, hydrogen ions, and oxygen. I'll come on to that in a moment. Here, the chlorophyll is positively charged. So what that means is the negatively charged electrons here are attracted to the positively charged chlorophyll molecules. This is an equilibrium reaction. So what it means is if you take something away from the right hand side, it upsets that equilibrium and it tries to compensate. So it then favours this forward reaction to try and balance it up. So indirectly, light is splitting water molecules. It's encouraging the forward reaction. And this is called the photolysis of water. So water doesn't absorb light energy directly. The light photoionizes chlorophyll molecules. That draws the electrons in and upsets the balance. We'll talk more about what is happening here a little bit later. Let's go back to our electron up here. This electron has got lots and lots of energy in it. And what happens is that in the membrane, and you don't need to know how many, are other electron carriers. And these electrons move between each of these carriers. And as they move between each of the carriers, they release a little bit of energy. And I'm just going to put that there for the moment. So there's a little bit of energy released as those electrons are passed between those protein carriers. Eventually, those electrons may come to another chlorophyll molecule in the membrane and are passed, so we'll just put these here like that, are passed to that chlorophyll molecule. That then is also able to absorb light energy, and the electrons now are in a much higher energy state. They possess a lot more energy, and eventually they will move to other molecules like so. And as they move, they still release energy, but the real energy that we're talking about is in this bit here. So what have we got left? Well, at the bottom here we've got hydrogen ions and we've got oxygen atoms. So that's an oxygen molecule, but there's only one oxygen atom in water. So to balance this equation up, it's actually half of that. It seems a bit funny, but that's the way we draw it. And what can happen is that two of them join together to form an oxygen molecule. These hydrogen ions now come along here and they are able to join together with the coenzyme NADP. And that coenzyme actually travels through the membrane and picks up the electrons and the hydrogen from here and are reduced. So let's go back to our little post-it note that I had before. Reduction is the gain of electrons, the gain of hydrogen as well, but we're more bothered about that. The reduction is the gain of hydrogen. Okay, so I think that makes sense. So what it means is now that NADP is reduced and we get reduced NADP formed. And that is a really important molecule for the next bit. Now, also in the membrane are some other protein molecules. One of those protein molecules goes all the way across the membrane, and that is a hydrogen ion pump. And what that can do is to move hydrogen ions from the stroma and actively pump them into the thylakoid lumen. Okay? And so this is active transport. Now, for active transport to take place, we need energy. And that energy, well, we've got some here. So the energy that was released from these electrons moving along their transfer chain is transferred to there. And don't worry about how it does it. In fact, these proteins are probably right next to these, because remember, the proteins in the fluid mosaic model are moving around, so they're probably closer together. But don't worry about that. So what we have is lots and lots and lots of hydrogen ions being pumped into here. And that creates inside a high hydrogen ion concentration. And outside, Therefore, we have a low hydrogen ion concentration. So now we have across this membrane, which if you remember is impermeable to hydrogen ions, a concentration gradient. In fact, we've got an electrochemical gradient. So here we've got some potential energy. These hydrogen ions want to move out down their electrochemical gradient. Electro because they're charged, so there's lots of positive charges here which will be repelling each other, and concentration because there's more of them here than there. But they can't get through here. However, there are other structures in the membrane, and the one structure that we talk about is a channel, and this channel allows hydrogen ions to pass through, and so hydrogen ions will be able to move down their electrochemical gradient out into the stroma. So what? Well, as they move, they lose energy. They're going down their electrochemical gradient, so they lose energy. And on the end of this channel is an enzyme. And that enzyme is called ATP synthase. And what that enzyme does is this. It uses the energy that is being released by the hydrogen ions flowing down their electrochemical gradient to join together ADP and inorganic phosphate to make ATP. So now we have a system where light energy is being transferred to ATP. And of course, this process on the right hand side here is chemiosmosis. And this process on the left is the light dependent reaction or reactions or stage. Okay, so what we've ended up with is this. We've got water coming in, we've got oxygen being released, and we know that water is on the left hand side of the photosynthesis equation, oxygen is on the right hand side. But we have two important molecules coming out. We've got reduced NADP and we've got ATP as well. So the question is, of course, what happens to these? Well, these are used in the light independent reaction. So in the light independent reaction, carbon dioxide is being absorbed, and then it's having hydrogen and electrons added to it to eventually form glucose. Okay, so how does that happen? Well, first of all, it happens in the stroma, and it is an enzyme catalyzed process. There's a series of reactions, but they are catalyzed by enzymes. And it's called the Calvin cycle. It's a cycle of events, but I'm going to squish it down along here because it doesn't really matter. Remember, this is a stroma, so all these molecules are floating around, bumping into each other, reacting and so on. So what we have in the stroma is a substance called RUBP, ribulose bisphosphate. That's got 5 carbon atoms in it. And that is able to combine with carbon dioxide molecules. Now, of course, carbon dioxide has only got 1 carbon atom in it. And what we get is RUBP, 1 molecule of RUBP combines with 1 molecule of carbon dioxide. And you get, instead of a 6 carbon molecule, you get 2 3 carbon molecules. And those are called GP, glycerate 3-phosphate. Okay. That GP is then converted into triose phosphate, which is also 3 carbons. But in order to do that, it needs some other substances. And these substances are here. It needs ATP, and ATP breaks down into ADP, an inorganic phosphate, to release energy for that process to take place. And you can see that these are now released and floating around in the stroma, and these could go all the way back to here and be used again. I won't put the arrow on because it'll confuse things more. But also we've got the reduced NADP. And the reduced NADP is also used in this reaction to give electrons and hydrogen to the GP. And so we get NADP formed. So that is how the two processes are related. Now, when we think about the light independent reactions, let's just think about quantities of each. Don't worry about how many GP, TP and RUBP molecules there actually are in the stroma. Just think that there's a quantity of each. But you do need to know the relationship between these 3 molecules. That 1 molecule of RUBP combines with 1 molecule of CO2 to form 2 molecules of GP. But there could be 1 million of them, 1 million of them, and therefore 2 million of them. And that would mean there's like 2 million TPs. Don't worry about what the actual numbers are. Just think that now in the stroma, there's a large number of TP, triose phosphate molecules, a really large number. Well, some of them will react together in a series of reactions called the sugar shuffle, which I think has been mentioned before, to form RUBP again. But very importantly is if you get 2 TP molecules, then those 2 TP molecules will react together to form hexose. In fact, they form hexose phosphate, and it loses the phosphate group to form hexose. Hexose, of course, is a 6 carbon sugar. And that is the basis of so many other molecules for plants. So some hexose might join together and form starch. Some might join together and form cellulose, for example. Some might join together and form sucrose, which would then lead into starch and other molecules like that. Hexose is also the basis to make amino acids. And from amino acids, we can get proteins. And we can also get fatty acids and glycerol. And from that, we can get lipids. We can also get nucleic acids. So basically, hexose is the starting product to produce lots of other biological molecules, different types. You don't need to know the details of most of these. You should be able to know that starch is a polymer of alpha glucose and cellulose is a polymer of beta glucose and so on. But you don't need to know the detail of how all these formed. But it is important that you know all of these, okay? And all of these together will lead to the yield or the productivity of the plant. And when you study energy flow and farming and agriculture, this is really, really important. So this photosynthesis isn't just a way of building carbohydrates, building hexoses, glucoses. It's a starting point to make lots of other molecules in the plant. And of course, to make these, other substances will be needed. So nitrates will be needed to make amino acids for the amino groups and so on. Okay, so hexose is C6H12O6. And now we can join together everything and summarize, if we think about our photosynthesis equation, which is 6O2 plus 6H2O gives C6H12O6 plus 6 oxygen. And that's our chemical equation for photosynthesis. And we've got each part now. Okay? We've got our water coming in here. We've got carbon dioxide coming in here. We've got glucose being formed here. And we've got oxygen being formed there. And so finally, this along the top is the light independent reactions. I hope that's been very helpful. Thank you very much.